So mitosis' job is to divide the nucleus, and the cytokinesis' job is to divide cytoplasm. We're going to also learn that there's a difference between cytokinesis in an animal cell versus that in a plant cell. All right, so if you want to, you can start a new note, but because this is referring specifically to mitosis and cytokinesis, you don't have to. However, what we see here are different aspects of chromosomes and DNA. So the first thing we see is a double helix or twisted ladder, spiral staircase. These are all nicknames of DNA. In the second picture there, we see DNA and histones. Now, DNA being wrapped around these little balls there is how all of our DNA is able to fit into small little cells that we have. If you were to take all of the DNA in one individual and connect it length by length, it would be very extensive. I don't remember the exact distance, but it's very long. Okay, we also have chromatin and supercoiled DNA. This picture here is one of an actual micrograph picture, microscopic picture rather, of a chromosome. Okay, so it's just enlarged. All right, let's describe this picture here of a chromosome. We can always, if we have many of these and we're trying to figure out how many chromosomes we have, we can actually look at the centromeres. The centromeres, these little circular parts, will tell us how many chromosomes we have. So this chromosome is made up of two sister chromatids. Okay, so a single length here, single side, is what would be called a chromatid. And again, the centromere holds the chromosomes together. Now, what telomeres do is that when copying happens, when DNA is being copied, there could be some lost in the copying process. So that's why telomeres exist, to help retain most of what is a part of the DNA in the chromosomes. So again, one of these single strands here is chromatid. On this side also, and together they make sister chromatids. All right, so this is a picture of interphase. We just went over interphase in the cell cycle, but this is just going over an overview really. It's not a part of mitosis. It's a part of the cell cycle. So don't include it with mitosis. However, we do see the structures. We see the DNA all condensed here, or beginning to become condensed. We have the centrioles, which are these items here, and the centrioles are going to be where the spindle fiber is attached to, so that the chromosomes can line up on the spindle fibers. All right, guys, 4 phases of mitosis. All right, we have prophase, which is the initial phase, okay? And in prophase, what's happening is that all the chromosomes are kind of condensing. So all that genetic material are starting to form chromosomes. And then the spindle fibers, you start to see those things too. So why are the spindle fibers important? Because that's where the chromosomes are going to attach themselves to, so that they can equally divide to the 2 new cells that are going to develop. After prophase, we have metaphase. Now, in metaphase, the chromosomes are going to line up along the equator. The centromere is how they attach. Now again, the reason why they line up in the middle is so that when it's time for them to separate into sister chromatids, you're going to have two equal amounts of chromosomes on either side of the cell. And that's why they line up in the middle. If they didn't line up in the middle, then you wouldn't have an even distribution of chromosomes when it's time for the cell to divide. All right, the third phase is anaphase, and this is when the chromosomes are now going to separate into sister chromatids. And then they're going to be pulled to the opposite poles of the cell. So they're going to go towards the centrioles. Okay, these 2 things, the centrioles. So at this point, we are beginning to prepare for the last 2 phases. Okay, or at least the last part of mitosis. Okay, which is telophase. That will be the next phase. Our last phase of mitosis is telophase. With telophase, we can see 2 distinct nuclei. We can also see the chromosomes begin to uncoil. So now they're going to be going back into their non-condensed, uncoiled form. But by the end here, we are going to have 2 new distinct cells. Okay, lastly, after telophase, we have cytokinesis. Now, cytokinesis is not a part of mitosis. Okay, there are only 4 phases of mitosis. However, it is a part of the cell cycle. Remember, interphase, mitosis, then cytokinesis. Now, it's important to understand that there's 2 different ways that cytokinesis happens in a plant versus an animal cell. In an animal cell, the cytoplasm pinches in and 2 new cells form. What prevents the cytoplasm from pinching in a plant cell? What structure?
The cell wall.
Yes, Vincent, the cell wall. The cell wall prevents it. Therefore, what has to happen in the case of a plant cell is that the cytoplasm is not going to pinch and separate the cells, but a cell plate forms. A carbohydrated structure forms in the middle that divides the 2 new cells in a plant cell. All right, guys, 4 major stages of the cell cycle. We have interphase, which has 3 parts. Gap phase, where your organelles, all the cells' functions are going to grow. Then we have the synthesis phase, and that's where DNA is going to be produced and additional growth. Now remember, you're getting ready for cell division. So 1 cell is getting ready to become 2. After that, we have mitosis, which has 4 phases. And then lastly, we have cytokinesis. Okay, so these are the 4 different parts that make up the cell cycle. And it's important to understand that the 4 parts of the cell cycle are different from the 4 phases of mitosis. So again, we have 3 parts: gap 1, gap 2, synthesis, and then we have mitosis. Now, if everything works according to plan and it passes all the checks and the cell grows large enough, then what will happen is that, and the DNA is not damaged, then it will go into nuclear division, which is what mitosis is. And after nuclear division, then we will have division of the cytoplasm, and that's what cytokinesis is. So as we see here, cells don't have the same time frame upon which they're going to divide. Notice how intestinal cells take the longest on this chart. What we don't see is nerve and brain cells, because they spend a lot of time in G0. They don't divide very often, nerve and brain cells. And that's why it's so debilitating to have one of those kind of injuries, a spinal cord injury or a brain injury, severe brain trauma, because those cells don't divide very often like other cells do. Okay, so different cells have different rates of division, and cells are constantly in one phase of division or another. Okay, so it's not like it's going to call on, all right, now it's time for number 459, and then number 460 is next for division. It doesn't work like that. Each cell of our organelles has different rate of division. All right, so the most important thing here is not to say, hey, well, cells aren't squares. Why do they have its square shape? Well, because when we're going over surface area to volume, when you have length, width, and height, it's easier to illustrate that using a square, because that's what you need in order for us to get that information. But the most important thing here is to show us that smaller cells have a larger surface area to volume ratio. Okay, 6:1 is greater than 2:1. What does that mean for cells? Well, it means that smaller cells are more efficient. Smaller cells can get information into and out of the cell membrane faster than a larger cell can. So yes, that smaller cell works a lot faster, a lot better. Things can go into and out of a lot quicker than it can with this cell. With cell, structure equals function. So each one of these cells has different surface area to volume ratio and functions differently. Okay, so structure equals function. Each has a unique shape that allows it to do its job inside of the organs or organelles that they're present. Okay, so here we have muscle cells, we have red blood cells, and then we also have nerve cells as well.